probably most of you know, and possibly not everybody, the sex ratio in autism is about 4:1, so it is dominated by males in terms of these diagnoses. The prevalence in the United States, the most recent data are 1 in 88, and that's 1 in 58 for males. And then the third bullet on this slide is about the, there's been a history in autism going back several decades where parents were blamed for their children's condition. It was attributed to bad parenting, a cold style, failure to bond with the child. And I think that not only is that totally wrong, and we really have a very strong biologic basis now that we know about aberrant brain development, and it began with some of the postmortem autopsy studies, and then those were supplemented by MRI studies, and that actually go back to the 1980s. Nevertheless, this myth that it was really a bad parenting that led to autism it has left a legacy, I think, of distrust between the biomedical community and the parent community, and I think that's something we need to be aware of in all of the work that we do, and how myself as a scientist, how I relate to the communities, to maybe take the extra step to make clear that this is not something that parents should be blaming themselves about. And in a way that comes up because I am looking at exposures that we believe may be modifiable. But if people didn't know, they can't really be held responsible. So we do know that there is aberrant brain development. We see it in these studies where the autopsy studies, which showed an absence or a loss of Purkinje cells in the brain. We see it in how electrophysiology studies map the electrical activity when people are doing various tasks and so forth. So, but what's the root cause of that aberrant brain development? So certainly we know that genes play actually a pretty strong role, and these can be everything from syndromes like Fragile X syndrome, Prader-Willi syndrome, Angelman syndrome, and so forth. There's also some single genes that seem to increase risk quite a bit. And then some of the most recent research is on copy number variants, where segments of DNA have sometimes been repeated erroneously in the transcription, so that people might have 10 copies where they should have had 1, or 30 copies, hundreds of copies even, or no copy at all. So deletions and duplications. And that's an area of very, very active research at this point in time. But in addition to genes, we know that there's also the question of what we breathe, what we eat, what we drink, the medications we take, the products that we use around our homes, and the products that we use on our bodies and our hair. So a wide range of environmental factors. And I'm going to also include in environment pretty much everything that's not genetic. So that will include medical conditions, as the maternal environment, the maternal uterine environment, becomes that environment in which the fetus spends its first 9 months. So just another point about the genetics and the environment is that the original heritability estimates were much higher. They were at about 90%, and even today you still hear people talking about 90% genetics, or 70, 90%, or 100% in a recent article in Nature, which had to go back to 1985 to find a single study that came out with a 100% estimate. But the most recent and the largest and most rigorous studies have put the estimate at somewhere up to between 35 and 60%. And one of those studies I've cited here, there was another one that came out about the same time. These were well over 100, between the two studies, it was about 300 or maybe even more pairs of twins that were studied to come up with these figures. So we know that environment really actually does play a pretty significant portion. And not only that, these heritability estimates don't really do a very good job of separating out purely genetic from gene environment from purely environmental cases. So the interaction between the two, and that may well be most of the cases where some genes contributed and some environmental factors contributed. So that's what I call multifactorial causation, where it's multifactorial not only from one person to the next, but even within an individual. And these little circles are called, sufficient pies or sufficient causes. So each pie might have anywhere from 2, 10 different factors, and each of those factors contributes, so that if you remove any one of those causes, any one of those factors, whether it's a gene or an exposure in the environment, it won't be quite sufficient to cause autism. So sufficient means that it takes every one of those to be sufficient. So here's an example. Maybe there are, A and B might be a couple of genes, C might be a pesticide exposure, D might be a metabolic condition of the mother, and that combination in some children may be sufficient to cause autism. This is another example, might be household chemicals. I'm putting up things that I'll be talking about a little bit later. And not only that, but these multiple causes have to unfold over time. So, for example, here's the E, which actually I think was the nutrition in the previous slide. So maternal nutrition maybe plays a really important role around the time of conception, but it may not actually play nearly as big a role, let's say, as you get closer to birth. So timing also matters in this model. So pregnancy is actually going to be the focus of much of my talk, but towards the end I will refer a little bit to postnatal exposures. But pregnancy does seem to be a really critical time period for lots of things that are developing in the brain. Nevertheless, the brain does not stop developing at birth. There are myelination, for example, continues on for another couple of years postnatally. So this is not to say that there are no factors that operate after birth. There probably are quite a few. They just have not been studied as well. So I came into the field really in about 2001, 2002. 2001, we were writing our first grant, and I reviewed the evidence. And as I go through this, I'll tell you a little bit about what we knew in 2000, what we knew by 2010. So definitely, it was well appreciated that genes were important, although CNVs, the copy number variance, was not yet on the radar in 2000. It was by 2010. Environmental pollutants, I'll be talking more about both air pollution and metals. Household chemicals, in 2007 were the first papers that I know of regarding household chemicals, and particularly pesticides. Two different studies. One actually was household pesticides, the one was residential proximity to agricultural fields where pesticides were used. And both of those studies showed either a higher risk of getting a diagnosis or a higher score on the pervasive developmental disorder not otherwise specified scale within PDD total on the aberrant behavior checklist. So there were measures in both of those studies suggesting that some class of pesticides might be associated with those outcomes. I'll talk more about vitamins and nutrition in a few minutes. And microbiological agents actually is something we knew about even in 2000, because in the 1970s there was an epidemic in the United States of rubella, and among children who were congenitally exposed there was a very high rate of autism, and we're talking 50 or 100-fold higher than the background at that point in time. Then there's been a literature on gestational exposures, conditions, perinatal conditions, maternal obstetric conditions. I'll talk a little bit about that. Fertility treatments have been of interest, particularly because these came into increasing use during the same time period that there's been an increase in autism. And then there's other medical interventions also that have risen, and I'll talk. I won't be talking more about these two, but the data on the fertility treatments has been inconsistent, so that's not really clear. And we have a paper that should be coming out very soon. Hopefully, we're in the revise and resubmit stage, in which we did a pretty detailed analysis, and we're not really seeing much of an association. So if there is one, it's probably a weak one. Other medical interventions of concern include ultrasound, and fetal ultrasound. It's not a radiation, but it is a thermal dose, thermal and mechanical, to the fetus. And around the time that the rates began to go up rapidly was also a time when the technology changed and new equipment came out that allowed for much higher doses to the fetus. So this is an area, it's actually very difficult to figure out how to study this, but I think it's certainly one of concern. And there are some animal models that suggest that that thermal dose could be enough to actually alter brain development. And then medications, I think the strongest data right now is about SSRIs, selective serotonin reuptake inhibitors, which are in fact prescribed for people, for example, with epilepsy. And that raises actually really important questions, because how do you control mother's epilepsy during the pregnancy and protect the fetus at the same time? So there are some really difficult issues here. And then modern technology, maternal reproductive characteristics, lots of questions have been raised, and every time I give a talk I get a few emails of people saying, have you thought of this? Have you thought of that? And often those are really good suggestions. Sometimes they're things that have been talked about already a lot in the literature. So a couple of words about time trends. And we did an analysis in California comparing births from as early as 1990 to 2001. And we found that the autism incidence by 5 years of age rose about 7-fold during that time period. So really a very dramatic increase in diagnosed cases. So of course, one of the questions is that really just more awareness, more clinicians who are qualified to make the diagnosis, or is it a true increase? And we tried to get at that by looking at some, what we might call these artifacts. So there was a change in DSM criteria during that time period. And from what we could tell, that would have explained perhaps a 120% increase. And what we're actually looking at is a 600% increase, because we started at 100%, we have a 7-fold increase, so it's 600% if you just look at it in percent terms. The trend towards younger age at diagnosis, which certainly has been going on, also explains some, but very little, 24% rise. Broadening to include milder cases. So this would be not necessarily the definition, but how this clinical practice actually happening out in the real world. Have people been more loosely applying this definition? And that seemed to account for possibly as much as 56% rise. And then advancing age at childbearing, people waiting till their 30s and even 40s to have children, seemed to explain maybe about 4%. So even though there is that trend, it's actually still the vast majority of people are having their kids before the age of 30. So the total from this is 204%, and we're trying to explain a 600% increase. And as you can see, we're a little bit of a ways from explaining it all. And that doesn't mean there aren't other possible things that are going on that just relate to how well it's being, the awareness. But it does seem as if some of this increase probably does have it may well be real. And as a thought experiment, I always like to throw this out, which is that what are the other conditions that have been rising during the same time period? We see asthma has been going up for several decades, obesity, and that's not an ascertainment issue. We can actually measure that really quite well. We have an objective standard, weight over height in a certain formula. Diabetes has been rising, ADHD, and several other mental health disorders, some of which may also be due to better diagnosis. So is there some sort of a unifying explanation? Is there some common cause, some changes in our environment that are actually partly responsible for these very diverse kinds of conditions increasing? So that's kind of all introductory material, and now I'm going to talk about the childhood autism risk from genetics and environment study. This is CHARGE. It's taking place in this area, and here at the MIND, actually, MIND Institute in Sacramento. And the goals are to identify the causes and the contributing factors to autism. So that's really the primary thing we've been working on for the last 10 years. Environmental exposures and a little bit touching on genetic susceptibility and their interactions of genes and environment. We also have a goal of trying to identify subsets of autism that might share some sort of common cause, but are characterized, behaviorally, perhaps immunologically, metabolically. We haven't gone very far down this route. We started doing some work in that direction, and we've just been so busy with all the other environmental factors that we have a lot of work to do on that. So I probably won't be saying a whole lot about the subsets. So when we sat down to make a list of what should we study, and having looked at the literature and found that very, very little was known about autism at that point in time from the non-genetic causes, first thing I put on the list was pesticides. The reason for that is that pesticides are designed to be malicious, right? The purpose is to basically kill lower species, usually insects, but it could be rodents, it could be mammals, and so that's how they're designed. So that's really what they're good for. And many of the biological activities that they have, the toxicities they have, are relevant. Maybe they're not relevant because the dose is too low, and the organism, we humans we have lots of resilience, but nevertheless, that makes them very high on my list. Metals also because they are known neurotoxins, and of course there's concerns about mercury, there's concerns, manganese is one that hasn't been studied very well, but is a very strong neurotoxin. And then many other pollutants, some of which I'll talk a little bit about. We were concerned, of course, about viruses based on the rubella epidemic, and also some data suggesting influenza could play a role. Medical procedures, I mentioned already, pharmaceuticals, and nutritional factors. So this was kind of our starting point for the study, casting a very wide net, and then looking at ways that we could actually measure and assess those exposures through the collection of biospecimens and interviews with the mother, and also linkage to databases that relate things like air pollution or use of pesticides commercially to where the mother was living. So these are just some of the arrows that can be made here, and we've looked at some of these arrows and some of them not yet. For example, the teeth that can be measured in which you could measure a variety of chemicals, where we've started asking our mothers after they participate in CHARGE, and the child starts to lose their baby teeth, could they bring those deciduous teeth in, or send them to us? We give them a little envelope so that we can use those to track actually the very early life exposures, because the dentin of the teeth gets laid down in utero, and so you can really have a window into very early life exposures that way. The newborn blood spot is also a really wonderful thing that we, where's that, up here, that we have access to, because the state of California stores extra blood from the heel stick from which they do newborn screening. And those stored specimens are available to researchers who can apply to access them for particular studies. So we've been able to obtain, right now we have actually close to 1,000 or more than 1,000 newborn blood spots from participants in the CHARGE study. This is the study design. It's case control sampling. I'll talk about that in a second. Population-based recruitment, so we're not relying on one particular clinic that might have some specialty issues and special populations. We're really casting a wide net. We use the California Department of Developmental Services DDS database to recruit, and we work through the regional centers as well. There is a standardized protocol that we follow for the diagnosis. And these things are not necessarily true in a lot of the research that's out there. So I think we're really trying to be as absolutely rigorous as we can. And then we're linked to a bunch of laboratories of immunologists and molecular biologists, and trying to use the latest technology to analyze specimens that we've collected. So we have, case control means that you have people who have a condition and people who don't. And rather than just starting with people who don't and then following them forward in time, which would be what we call a cohort study, case control study is quicker to do, but sometimes trickier to do. So we have children with autism as our major case group, and then we have another group, which are children with developmental delay who don't have autism. And that group can be compared either to the children with autism, or they can be a case group themselves. And then the controls are these children who we select by using the birth files, so they really are also population-based, just like the cases, and we match them on age, gender, and geography. So those are the children that we've been looking at, these three groups. All of them have to be between 2 and 5 years of age, 24 to 60 months, born in California, so we can get their newborn blood spot. Living with a biological parent, and that parent needs to speak either English or Spanish, and they need to reside in our catchment area. And the catchment area is basically, it's these 4 regional center catchment areas around Sacramento, the green, the pink, a little bit of the gray, and this yellow area here. And then in the early part of the study, we also had a site in Los Angeles, but we weren't able to maintain that. At the clinic visit, we do the Autism Diagnostic Observation Schedule, ADOS, and the ADIR, which is the interview with the parent. So we're basing our diagnoses on both of these instruments. There's also a medical examination and a history, and you can see Robin here with a child. And we do a broad assessment of cognitive and adaptive function, and also deviant behaviors, also comorbidities. Then we do a lengthy interview with the mother, in which we ask her lots of questions about her pregnancy history, her medical conditions, lifestyle factors, household products, some dietary questions, and so forth. And we collect specimens from as many members of the family as are willing. And then we get permission to obtain the medical charts. So that's how we collect the data and what we collect. And our slogan to the parents is, "Don't you think you should be in charge?" Okay, turning to results. So we were interested in looking at metals. There was a lot of concern, and a number of parents had come to us to say that they'd heard that their children were loaded down with metals, and was that really true? And should they be chelating their child, for example? So we determined whether the current blood mercury levels in children with autism or ASD, as compared to the typically developing children, were different. So a little background on mercury. Mercury can come in 2 forms, inorganic and organic. The inorganic is usually the exposures are from some sort of occupational industrial type of work, or from dental amalgams. And the actual amount that you get from having those amalgams in your mouth is actually pretty low, but at the time they place them or they remove them, there could be a spike. You'll hear a little bit more about that. So organic mercury is what you get from eating fish. And so fish and seafood, that's the biggest source for most of us. And then there's also small amounts in vaccines and in some of these products like nasal sprays and contact lens solutions. although they've taken it off the label, but it was on the label when I first got involved in the study and took a look, and I've been wearing contact lenses for decades now, so. Okay, so these are the data, and what you're actually seeing here is that, so this is going sort of from low to high levels of mercury. These are the units in log micrograms per liter for the technical people. And then on the y-axis is the percent. So here you have, a very small percent who have the very low or who have, say, the very high. And what you actually see is that the typicals seem to be shifted a little bit to the right. So the typically developing children actually had higher levels, not lower levels, than the ASD children. And the same for the developmental delay was more similar to the ASD, shifted a little bit more down, right? And the reason that turned out to be true was that the ASD children were less likely to eat fish, and the ones who ate fish were eating fewer fish meals per week. So the picky eater part maybe came into play here. And that we had to think a lot about, well, how do we analyze those data then? So what we did was we adjusted for their fish intake, and we fitted a statistical model in which all of these factors are all part of the model, and this has been published. And we showed that, in fact, you can see that fish, almost any kind of fish, really pretty strongly increases your amount of mercury in the blood, doubling, almost doubling here. Whereas the difference between the autism or ASD group and the typically developing children was like 4%, not significant at all. This high p-value means it's not significant. And then the other thing I want to draw here, and by the way, even after adjustment, there was still a lower level in the developmental delay group, and we actually don't exactly understand why that is. But we do see that if they had these mercury amalgams in their mouth, and these were young children, so not that many did, but if they did, that didn't do it, that didn't change their blood mercury. But if they had them and they also either chewed gum a lot or had a habit of grinding their teeth, that seemed to lead to some sort of release, low level, only a 19% higher, but some release of the mercury. And that's, that is, plausible. It's quite consistent with what we know about those amalgams. So the bottom line then is that the children with ASD do not have the higher blood mercury, and they don't really have lower blood mercury either once you adjust for their fish consumption. I want to add here very emphatically that the addition of a little bit of mercury from fish is actually pretty strongly outweighed by the benefits of eating fish during pregnancy and in early life for children, because fish, you've heard this phrase, brain food. It really is true, and in many studies now, they are able to show that children who have higher mercury might have lower cognitive levels, for example, but if they have been eating fish, they have higher cognitive scores. So it actually is truly beneficial, and there's a lot of theories about in evolution that when people started eating fish, that was sort of when our brains started to grow. So it's really, they're critical nutrients, omega-3s in particular, in the fish. And there are fish that are low in mercury, and there are a lot of websites out there that you can go to look up what those species of fish are, and a lot of interesting work that's been done in Hawaii, where of course they eat fish 3 meals a day on that question. But I also want to add that this does not address the question of mercury as a causal factor, because we're looking at the mercury in the children who already have the diagnosis, and mercury has a half-life of months, not years. So in order to address that question, we have to look at earlier measures, and we have been doing that on the hair, the baby hair, and the blood spots to try and get at early life exposures. And I am have to go back and kick the graduate student who did his dissertation on those topics and get him to publish that, because I would like to start talking about those results as well. Okay, air pollution is another issue that has come up. It's a source of a lot of different pollutants. And a lot of it in our current years comes from traffic, from freeways and from heavy traffic areas. In other words, the vehicle exhaust. So it turns out that distance to freeway is a nice way to look at this. So we were determining whether the mothers of cases were more likely to live closer to freeways and therefore have greater exposures to ambient air pollution than the mothers of the typically developing children. And these are the results, and I'll talk you through this slide. So we're comparing to basically the 50% of the children in our study who were farthest from freeways, and that turned out to be about 9/10 of a mile or more. So that's what we call our referent group. And then we had 3 different levels of exposed. These are people who are 4/10 to 9/10 of a mile from the freeway, 2/10 to 4/10, and then less than 2/10 of a mile. So this is the closest group, and you can see they have an odds ratio of 1.86. So that's about getting close to a doubling of risk. An odds ratio of 1.0 is really what our referent group is, and that's what we're comparing back to. So those people living quite close to a freeway, less than a fifth of a mile, were at higher risk. And there was not, however, some people say, well, was there kind of a dose response as you got closer? Did it get higher and higher? And it doesn't look like it. It looks like these 2 groups are very close to 1. And it turns out that that's actually very plausible because the levels of particulates coming from vehicles are extremely high in that first 300 meters, which is about 2/10 of a mile, and then it drops off. It's just about background level, as if you were, it's really not that much different from these people who are living further away. The nice thing about this particular exposure is that we're basing it on something that people aren't having to report. Well, they did have to report where they live, but they gave us addresses. They didn't know, what we were going to do with those addresses. So it's not asking people, remember, how high was the air pollution when you were pregnant? That's not the way we collected these data. So it's a very objective measure. And there's lots known about the constituents of air pollution, these benzoate-pyrenes, the diesel, the ozone. In animals, they do induce neurodevelopmental deficits. And then some of these compounds are PAHs, polycyclic aromatic hydrocarbons. They are nasty substances. This is one of them. They can cause inflammation, endocrine disruption, lots of different sorts of toxicities. Which is why, we do have regulation to reduce, emissions and keep our air clean. But then I have to ask the question, so what about cigarette smoking? Because, in fact, the constituents of cigarette smoke are very similar to what's in traffic pollution, traffic-related air pollution. Many of the same compounds. But the cigarette smoking literature right now is very, very confusing and inconsistent in relation to autism at this point. I think part of that may be because for that, we often have to rely on asking people, did you smoke? And a lot of people are not very quick to admit that they smoked, especially during their pregnancy. So I think the quality of the data in those studies may be in question, but there does need to be a lot more work to figure out what the story is in regard to cigarette smoking. And is there really a discrepancy here? So turning now to maternal nutrition, and this is, I think, one of the areas where we've made some really terrific progress. I think it's really one of the most shining pieces of our research right now. So just a little background on folic acid, which is required for DNA synthesis, DNA repair, and DNA methylation, which I'll talk a little more about. And we, in fact, ask women or recommend that women take these prenatal vitamin supplements before they get pregnant, the reason being that in the last 20 years or so, we have established that folic acid is critical for the development of the neural tube, which is sort of the beginning of the brain, right? And supplementation can actually protect very strongly against those neural tube defects. We also have fortified our cereals with folic acid, and there are a number of studies that have shown a declining prevalence of these kinds of birth defects since we started doing that. So this is a slide showing the results from the CHARGE study, in which we looked at what mothers said that they took at different time points, starting 3 months before pregnancy, the -3, -2, -1, and going through each month of pregnancy. And what you can see is that the case mothers and the control mothers both were, over 80% of them were taking these prenatal supplements by month 2 and beyond, okay? But before month 2, month 1 of pregnancy, and those 3 pre-pregnancy months, there's actually a significant difference, and you can see that the case mothers were less likely to be taking these supplements. They were less likely by about 40%, and that's where this odds ratio 0.6, it's the taking of the supplements actually reduces your risk. So we were looking at air pollution, it increased the risk of an odds ratio above 1. These are below 1. It's a protective effect from taking the supplements of about 40%. So this was published in 2011. And we also looked at how much folic acid they were getting through diet and supplements, and you can see that this actually does show what we call an incremental kind of dose response, where the more you took, the more protected you were from autism in the child. And very, very highly significant trend. This finding is actually supported by some other pieces of information, things that have been published by others, including a study of interpregnancy intervals. So this is the interval between one conception and the next conception. And if it was less than 12 months, those children were at extremely high risk, 3.7-fold higher risk. If it was between 12 and 24 months, it was about a 2-fold higher risk, and this is comparing to more than 36 months from conception to conception. And then 24 to 36 months, slightly elevated here. So one of the aspects of pregnancy is that it very much depletes maternal nutritional reserves, and it just really, maybe it takes a good 36 months conception to conception, which is actually like, 27 months from delivery to the next conception, for the mother to really rebuild that reserve, potentially. That's one possible explanation for this finding. There's also now been published in 2013, just earlier this year, a group in Norway actually replicated our findings. And that was a study, a very large study, that was conducted in which they had enrolled about 85,000 pregnant mothers and followed them to a mean of 6 years. They recruited during pregnancy, and they asked them at 18 weeks about their prenatal supplement use, okay? And they found a 40% reduction, and this is sort of this, it's a slightly different window of taking it than we looked at, but it's close. They again found that 40% reduction. So now 2 studies with almost identical results, and I think it's really, this is really, really strong evidence, because these mothers were asked before they knew any outcome of their child. And then just a qualifier here, which is to say that the supplements contain things other than folic acid. They also contain other B vitamins and iron, and those may also play a role. And then I'm going to talk just a moment here about a gene-environment interaction with regard to nutrition. So this is something that nutritionists are very familiar with. It's the folate pathway here, where folic acid comes into the body, it gets converted to folate with the help of the enzyme that this gene codes for. That folate then goes into this next cycle and contributes these methyl groups, which are carbon and 3 hydrogens, that are really critical for the functioning of DNA, RNA, and neurotransmitters. So this is the cycle, and that transmethylation, transfer of the methyl groups, is regulated by this COMT gene. So here's an example of what we call gene-environment interaction. And so this is the not very efficient transmethylator genotype. And so these are the efficient methylators. And so we have efficient methylators who also took their supplements, and they're the referent group. So if you have inefficient methylation, but you did supplement, your risk for autism was about 30% higher, a little bit of an increase here. And if you didn't take the supplements, but you did have efficient methylation, then, or transmethylation, then you also had an increased risk, but a little bit higher, 1.8-fold. However, if you're an inefficient transmethylator and you didn't take the supplements, those children were at 7-fold higher risk for autism. So this is what we call a synergistic effect between the genetics and the environment. And something sort of similar with one of those other genes I pointed out, the MTHFR, which was the folic acid to folate conversion. And here, this is the inefficient converters had a reduction. So this is the overall reduction in risk. This is in that group. It's an even stronger reduction. Same if the child had the inefficient conversion. And if they both did, then it's an even stronger effect. So that's the group that seems to really need to be taking their prenatal vitamin supplements. Okay, maternal metabolic conditions. The fetus depends, of course, on the child for nutrition and lots of other things. And maternal metabolic conditions, in particular, insufficient regulation of blood glucose, can operate through a number of different pathways and eventually result in neuronal damage. And I won't go through these in great detail, but clearly the fetus is influenced by the mother's glucose level, and this can lead to hypoxia. There are immunologic changes that also can be stimulated, that also these cytokines, pro-inflammatory chemicals, can cross the placenta and eventually affect the fetal brain as well. So there are a number of ways in which the maternal metabolic condition can actually influence child brain development. So one of my graduate students, now doctorate, Dr. Paula Krakowiak, did an analysis of these conditions. These are 3 metabolic conditions, and the combination of one or more of them found that there was a higher risk for autism in children of mothers who had either obesity, diabetes during the pregnancy, or hypertension. And there was also an effect on developmental delay without autism, as you can see. And then another finding that she had was that among both the ASD children and the non-ASD children, there were changes. In particular, there was deficit in receptive language and in expressive language if the mother had actually, in this case, it was this analysis was just diabetes alone. And then there was also some deficit in the Vineland subscale on socialization. So several factors, and it applied both to the ASD group and the non-ASD group. And there are prior studies of maternal diabetes, and of course we know we get macrosomic babies, so clearly their bodies are being challenged by poor glucose regulation and hyperinsulinemia. So then there's another question though, which is what is it that influences these metabolic conditions? And I alluded to earlier this issue of what else is rising with autism, and these are conditions which have been increasing. So maybe part of the time trend is related to this increase in obesity, or maybe both of them are downstream from various chemicals in our environment, like bisphenol A and antibacterials, which have become very, very ubiquitous in our household products. Plastic water bottles for the bisphenol A. Half the soaps out there, advertise that they're antibacterial. These compounds, have an impact on our environment and our microbiome. And our endocrine system. So you do see in cross-sectional studies, remains to be seen in longitudinal studies, but there are strong associations between diabetes and some of these compounds. And actually some work done here at UC Davis by Kimber Stanhope, very interesting work on high fructose corn syrup, which has, practically completely replaced sugar in most, most processed foods. And for high fructose corn syrup is not treated by the body the same way as our more basic sugars, the glucose that we would get, say, from or fructose directly from eating fruits. And it actually does stimulate several of these pathways that lead to poor diabetes and those kinds of pre-diabetic conditions. A few words now about the immune connection, and we have been collaborating with two wonderful immunologists, Dr. Paul Ashwood and Dr. Judy Vandewater, and I should put they have many, many newer publications. These are all from about 2008 to 2010, but they found a lot of differences between children with autism and children who are developing typically in sort of basic functioning, immunoglobulin levels and productions of cytokines and this TGF-beta, which regulates the immune response, for example. But one of the most striking findings has been something they detected in the mothers of the children with autism, who in about 6 to 12% of them produce a particular set of antibodies to proteins that are found in fetal brain, but they're not there, in adult brain. They're there in the fetus, and they're not in fetal bone or fetal muscle. They're very specific to fetal brain tissue, and mothers are actually making antibodies to those proteins in this small subgroup here. And in studies now, we started out just a few dozen, but by now we've done hundreds. We have never found this particular pattern in any of the mothers of the typically developing children. So it seems to be very specific, although not fully sensitive, because it's only 6 to 10% of them, of the cases, of the ASD case mothers. But this raises some really interesting questions. We don't really know, did the child's autism somehow influence the mother, or did the mother making these antibodies influence the child's brain development? That's, it's very unclear how this is in fact causing autism or not causing autism. But it does raise a larger question that environmental chemicals could influence neurodevelopment indirectly through dysregulation of the immune system, and the immune system is very important in brain, and there actually have been papers out suggesting that autism, or at least some subgroup of autism, is an inflammation going on, a chronic inflammation in the brain, neuroinflammation. And along with that, we have looked at the role of infections and fever, particularly looking at influenza during the pregnancy. And we actually found that in our initial analysis, which is not shown here, we found that overall women who reported having a fever were more likely to the case mothers were more likely to report having had a fever in pregnancy than the controls. And then we presented that result at a meeting, and somebody in the audience got up and said, well, what about, if they controlled the fever with medication? So we went back and we analyzed it, and what we found is that those who had the fever and took anti-fever medication in the autism group, whatever association we'd seen was much more muted. So it was an odds ratio, as you can see, of 1.3. And the numbers after that 1.3 is kind of the degree of confidence. So we think it could be as low as 0.59 all the way up to 2.84, right? And then, however, if they had the fever but did not take anything to control that fever, you can see it's a 2.55, 2.5-fold higher risk. And the referent group is those who didn't have a fever and, of course, didn't take anything for fever. So that's, that was the finding about fever and influenza. And there are some other studies suggesting that prolonged fever can be a problem, so uncontrolled fever then. Of course, this is actually based on self-reports, so we can maybe aren't quite as confident of these findings as some of our other ones, but it is very consistent with some rodent work where pregnant dams were injected with influenza, or maybe not actual influenza, but a part of the virus that would stimulate the mother to respond as if she had an infection. And they produced in these rat pups this, reduced exploratory behavior and some changes in what's called pre-pulse inhibition. So things that really are relevant to what we can, what the behaviors of autism are, to the extent you could say, what would an autistic rat, look like. Okay, so just summarize the take-home messages from the CHARGE study. One is to take prenatal vitamin supplements before pregnancy, and the best is 3 months prior to start them. Space your pregnancies apart. The best would be 36 months from conception to conception at least. Limit exposure to air pollutants, and I would suggest traffic, cigarettes, and other sources, wood-burning stoves. Reduce weight and control your blood glucose. Exercise moderately to maintain lower blood pressure. Avoid the flu or prolonged fever and take anti-fever medications if you do get it. And limit exposures to chemical pesticides. I should also put in here SSRIs if you don't need them, or consult with your doctor about SSRIs. And that really is pretty much what we know about environment and autism and the modifiable factors. So that has been our focus, is what can people actually change? What is it that's modifiable? Genes are kind of hard to do that with, because we want to really be able to intervene. And we've seen these rates going up and up and up year after year. My dream is that before I die, and maybe even before I retire, that will start to plateau, and then we're going to start to see those rates coming down. So that's really the goal of all of this research. Now, I just want to say as a caveat, and as just a short couple of minutes, I want to talk about the other study we're doing, the MARBLE study, that the limitation in this study is that some of our exposure measurements do require mothers to remember things well, and we know that mothers who have a child affected with something very severe may remember things differently, and they may remember things better, in fact. It may be that the problem in many cases is that the mothers of the typically developing children haven't spent a lot of time racking their brains trying to think, what might have gone wrong here. So they may not remember that they had the fever during pregnancy, or they may not remember, did they use, pesticides or what have you. So the answer to that is to conduct a prospective study, and I already referred to that Norwegian study that was prospective, and that's a large, 85,000, mom-child pairs. But we can do smaller studies if we have a higher rate of autism, and the way we do that is we take moms who already have a child with autism, we enroll them, and then we follow a subsequent pregnancy. And because of the strong familial risk part of it is the genetic component, some of it may also be common environment, that there's a relatively high rate of autism in families who have one child already. Most of the studies are somewhere around 15 to 18%. So this is the study we're doing, Markers of Autism Risk in Babies Learning Early Signs, and the CHARGE study, which has well over 1,000, somewhere around 1,700 children involved. This one is much smaller, of course. It's much more labor intensive because we don't just see them all once or twice. We see them time and time again to collect specimens and ask questions and evaluate the child at different ages. So these complement those case control studies. They eliminate that problem about the differential reporting because these mothers don't know what the outcome of this child will be. It also allows us to look at relevant time windows more closely. And so Marbles is one. There's another study called Early in similar design. And then there are these very large population-based studies. National Children's Study is what the NCS stands for, and MOBO, that was the Norwegian mother-baby study. I don't know what that means in Norwegian, but Okay, so then we actually have one, I'll just give you one little piece from the Marble study. So we were approached by Dr. Kleiman from Yale University about looking at the placental tissue, and we had been collecting placentas on the Marble's pregnancies. We work out with the delivery room to save the material, and then we process it and store them. And Dr. Kleiman was interested in these trophoblastic inclusions. So what you're seeing is, you know, there's a fairly early embryo, and this is the chorionic villus that they sampled for, you know, some testing. And then this is looking at these what are called trophoblastic inclusions, which is tissue that should have fused with the fetal tissue but didn't. And you can see this is a case, and then this is not a Marble's mom, a mom who's never had a child with autism but has had previous children. And the number of these inclusions, it turned out, was much larger in the case group than the control. So here, all of the control pregnancies, oops, wrong button, had 0, 1, or 2. And you can see that the Marble's mom spread out all the way. One placental tissue had 15 of them. So we don't know exactly whether this is, you know, what it actually, you know, is this a sign of something that was going wrong very early in the pregnancy, and how is it? Okay, so. Meaning of that yet to be determined, whether it relates to environmental exposures yet to be studied. So just a little tantalizing piece for research. And then I just want to say final words. I haven't really presented anything about postnatal influences, although we did look at some of the data on air pollution during the first year of life. There's a strong correlation between where people lived during pregnancy and where they lived postnatally. So we don't know. But we did see an association with the postnatal air pollution. There's also some intriguing data about breastfeeding possibly being protective, and one study that suggested that breastfeeding, or if you gave formula and that formula was fortified with DHA, then maybe that was almost as good as breastfeeding. But the formula fed babies where the formula was not fortified seemed to be the ones at higher risk. But that was only one study and not a very large or strong one, but definitely some interesting findings. And then there's the issue of post-diagnosis and what can you do. And of course, that's no longer prevention, that's actually treatment. But we are interested in that, and we actually will be hopefully looking at that. We submitted a grant, which we're very hopeful that we'll get funded, called the Recharge Study, that we will bring charged children back when they're 9 to 13 years of age. You know, this is started out as a case control study, but we can now follow those children forward and look at the sort of the overall natural life history course of autism from ages 2 to 5 up to adulthood. And so we're hoping that this will be the beginning of that whole portion of the charge study. And then just thanks to many, many people, I've tried to sort of highlight the people who have played a big role in some of the results that I showed you. And I think I may have failed to mention Dr. Schmidt, who did all of the nutrition work. And I think I mentioned many of the others here. And then the many staff who did a huge amount of work, from recruitment to interviewing to everything, and maintaining that relationship with the families. And Robin has been really very instrumental in making that work and that go smoothly, so people have a good experience in being in the study, even though we, you know, hammer at them for hours and hours on end. So thanks to the many families for both of the studies and our funding agencies, and thank you for listening. And you can contact me if you have questions. I do want to just make a plug for the studies that we are ongoing recruitment for both the CHARGE study and the MARBLE study, and if you yourself or someone you know might qualify for those studies, we'd be happy to send you materials and bring you and your friends and your clients and so forth into the research process.
The UC Davis Mind Institute was created in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other brain disorders are helping children achieve their fullest potential. Please visit our website to find out more about current studies, upcoming events, and how you can help make a difference.